Okay, so today we're going to talk about meiosis. But before we get into meiosis, I just want to review a little bit with mitosis. So, if you remember, mitosis is a form of asexual reproduction. And meiosis is now going to be a form of, or involved with sexual reproduction. With asexual, there is one parent. And with sexual reproduction, we're going to have two parents. Mitosis now, if you remember, we're going to start off with one parent cell, and we're going to wind up with two daughter cells that are genetically the same. Okay, there shouldn't be any difference unless if there was some mutation or something, but for the most part, they're going to be genetically identical to each other. We wind up with two cells. With meiosis, we're going to start off with a diploid cell, and we're going to wind up with haploid cells. Now I'm using a new term, diploid and haploid, which we haven't went over yet. So before we go further, let's explain what it means to be diploid and haploid. A diploid cell has the total number of chromosomes for the species. Okay, for the species. If you're and that's represented by 2n. A haploid cell has half the number of chromosomes. And that is represented by n. So if we take a look at this as far as humans, right, our total number of chromosomes is 46. So every cell in our body is going to have 46 chromosomes, and it's represented by 2n. If you wanted to find out how many chromosomes our gametes would have, our sex cells, well, we want to solve for n, right, because gametes are represented by n. So if you do this mathematically, and you solve for n is going to equal 23. So our sex cells, our gametes, should have 23 chromosomes in them. So that's where this expression 2n and n come from. Okay, so you see, you do need math for other subjects. So, and this is really important because the sperm is going to have n, or 23, and the egg is going to have n, or 23. And when the sperm and egg come together to fertilize or to unite or to form a zygote, it's going to bring it back to 2n. So that's why that is necessary. So let's take a look at meiosis and see how this happens. So here we have a cell. For this species, the diploid number is 2. So you have one chromosome that came from the father. I'm going to represent that in blue, and one that came from the mother. I'm going to represent that in pink. They code for the same genetic information because you have this is referring to two chromosomes or one pair. So if we take a look at the next step, what's going to happen is the genetic information is going to duplicate. So the chromosome that this individual got from its father made a copy of itself, and the chromosome it got from its mother made a copy of itself. These are called sister chromatids now. Sister chromatids. Oops. And what will happen in the next step is that once the copy is made, they're going to unite with it. They're going to combine. Okay? And it's going to form a tetrad. So a tetrad has 4 chromatids attached to it. And remember from mitosis that you have, the centrioles, and they're going to be attached to the centromeres of the chromosome, and they're going to pull apart. But what's significant here, what's really important, is that a step called crossing over is going to occur. And that's when the tetrads are going to actually intertwine with each other. And it may look like something like this. And they become all intertangled. And if you could imagine that now the centrioles are attached and their job is to pull and separate this tetrad, and you'll see that the where they're crossing over, where they're connected, genetic information could switch from the pink to the blue and the blue to the pink, and you may wind up with something that looks like this. Okay, so instead of having solid blue and solid pink, you have a combination. And if you have a sibling and you take a look at your siblings, or you look at someone else, if you don't have a sibling, you look at someone else, you'll notice that everybody in the family, they don't look exactly the same. And if they have the same parents, then why don't they look exactly the same? Because you think they're coming from the same gene pool. So why don't they look exactly the same? Because of crossing over. This is totally random, and every sperm that's produced by a male is going to have a different combination of genes, and every egg produced by a female is going to have a different gene. There's not going to be a different set of chromosomes. You'll never have two eggs from the same woman that have the same combination of chromosomes, and same thing with males. A male will produce between 200 and 400 million sperm for one ejaculation, and not one of them will have the same genetic information. Then what will happen, we're not done yet, because if you look here, this is diploid. One more cell division is going to take place, and they're separated into one gamete. So here we wind up with 4 gametes, and they are N, right? N equals 1. We're down to 1 chromosome. So if this was sperm, one immature sperm cell, one spermatozoa, is going to produce 4 sperm that are haploid. So what I want you to do is, for class tomorrow, is I want you to look at, in your textbook, spermatogenesis, and oogenesis. This is just fancy for saying meiosis in forming sperm cells, and oogenesis is just meiosis occurring in eggs. I want you to see how they're different. Okay, what happens differently between the two? Because meiosis is the same, right? What happens to the chromosomes is the same, but there's a couple of differences, and I want to see if you could come up with that on your own. Okay, and we'll talk about this more further in class tomorrow.